All right, thank you all for being here. It's good to see some people in person. And I'm pleased to introduce Dr. Chris Hakkenberg, who is a postdoc at Northern Arizona University. Chris is probably the person in academia that I've known longer than anyone else. Before I even started day one of grad school, Chris was like someone that I had met. We started our PhDs together in the same lab the same year, graduated in the same year, and so we've been friends and colleagues and collaborators now for more than 10 years, I think. So Chris got his bachelor's degree from Reed College in Chinese studies and a master's degree from Harvard in regional studies, East Asia, I think, is that right? Yep. Studying conservation practices and policy, particularly focusing on the conservation practices and traditions of Tibetan Buddhist monks, and then did his PhD in ecology at UNC. And so Chris is a community ecologist and remote sensor, studying things like forest community composition, biodiversity, and the relationships between ecosystem structure, diversity, and function, largely using remote sensing technology, including things like multispectral, hyperspectral, and LiDAR remote sensing. And then in addition to that, Chris has also done a fair bit of work on land cover and land cover change, work that we've collaborated on together, and has published in a large range of the most prominent ecology and remote sensing journals out there, including Ecology, Ecological Applications, Global Ecology and Biogeography, Remote Sensing of Environment, Remote Sensing, Photogrammetric Engineering and Remote Sensing, a lot of our major journals. So with that, please welcome Dr. Chris Hakkenberg, who is going to talk about biodiversity, forest structure relationships, theory, findings, and emerging applications. Oh, and another quick reminder, we're doing a happy hour at my house afterwards at 5:30. My address is 409 Brown Street. All right, take it away, Chris.
I'm gonna minimize this.
Thank you, Matt. Matt's right. We have known each other for quite a long time. Matt, you've always been one step in front of me, and so I try as I might to catch up. You've got me there. So good stuff. And thanks so much for the invitation. From Matt, also from Susan, who's just helped to facilitate this really cool trip, my first time in Iowa City, and it's a charming little town, to say the least. So yeah, today I'm going to talk about kind of one theme of my research, and that is this biodiversity forest structure relationship, and try to get into why it matters and how we can use this to help us better understand the planet, but also to be able to kind of contend with some of the global change issues which are affecting us, like habitat loss and climate change. So very quickly, hopefully not too redundant with what Matt said, but I'll just say some of my research that I've been working on recently uses field and remotely sensed data to examine forest community dynamics across scales in space and in time. And you can see some examples here of kind of how this spatial scaling works out as we go from the plot level all the way to the continental level. The kind of penultimate one over there, you can see there's layers. That's meant to depict change through time. So that's that kind of temporal scaling. And I'm kind of using these approaches to engage in these three kind of topic areas. So drivers of diversity, structure, as Matt said, so the individual forest, but over larger scales of the landscape, and then how ecosystems are responding to these global changes. But again, today, I'm just going to be talking about this focused topic of biodiversity and forest structure, which allows me to go a little bit deeper into my career. And so I'll be talking about some stuff I'm doing now, but I'm also going to be talking about some dissertation work. And some of the theoretical work emanates from this idea that diversity begets diversity. And in this sense, biodiversity, by which I mean taxonomic or phylogenetic diversity, so it's the number of species or the genetic diversity, how that is related to the diversity of structural types or the three-dimensional distribution of biomass in a forest ecosystem. So there are two different types of diversity. And the question is, are they related? And if so, why? And why does that matter? So this is not a new topic. People, prominent ecologists have been dealing with this for quite a long time. This is an old paper by the famous ecologist Robert MacArthur, where he looked at the relationship between bird foraging in different parts of the canopy profile of different forests, and then actually looked at different forests in different places in the Americas and sought to see how much he could predict the diversity of birds solely on the structural characteristics of those forests. And these are kind of these cross sections of these forests that gives an idea of where we're seeing more foliage, more branching, and what sort of species we would see and what the biodiversity is. So this is in 1961. There's been a bunch of work on the topic since. But today I'm kind of going to end this with a kind of forward-looking approach and how modern sensors and satellite remote sensing can help us to really push this original research that he did in 1961 into the 21st century. So this is a quick outline of what we're going to be talking about. Findings just based on field data, so no remote sensing. Moving on to remote sensing at the landscape scale. Then remote sensing and biogeographical theory at the continental, or at least the subcontinental scale here, the contiguous US. And then finally, those emerging applications that I alluded to a moment ago. So let's start with findings. Okay, this is a chapter of my dissertation where I sought to explore just the simple question is, does forest structure predict tree species diversity in this particular study area, which is the intersection of the Piedmont bioregion and the state of North Carolina. It's a little bit arbitrary, but it was a good way to kind of constrain this data set. And we use the forest inventory analysis data set, which is a data set which is across the United States, which allows us to have an amazing picture of what's going on with forest dynamics, ecosystem dynamics, and community dynamics. In this particular set, there were a little over 1,000 plots that we could use. We don't have the exact spatial location, but it doesn't matter because this isn't remote sensing. We're just looking at how these relationships in the field play out. And specifically, it's the relationship between forest structure and species diversity. So quickly, this is a glimpse of the distribution of these different types of forests in North Carolina. Some of the similar species that you see here in Iowa, and break these up between pine, mixed, and broadleaf, mixed being part broadleaf, part pine. And the second way to break these up is natural and artificial. And so this just kind of shows the distribution of those in relation to richness and two different measures of structure, maximum height, how tall it is, basal area heterogeneity. Just what that means is just the variance in the sizes of these trees. And the main thing I think that you see here is that these broadleaf, these are the forests where we're seeing a lot more size, structural diversity. We're also seeing more species there. Over there on your right, you can see a breakdown between the stand age and as well as the distribution of the total plots. You can see there's a lot more broadleaf, and those broadleaf tend to be a bit older, and those pine forests are younger, and some of those could be plantations that were planted relatively recently. So here are some of the individual metrics. I don't get too caught up in all the detail here. I'm just showing this really quick to say that with diversity, there's lots of ways to characterize diversity. I'm going to focus on one kind of graspable one, which is species richness. And that just means how many species are there. So I'm not going to get into these other ones, which weight various other factors more or less. With structural metrics, again, there's hundreds of metrics out there. here I kind of focus on these summary statistics. So, maximum height, mean height type of thing, the density, how many stems are there in a plot. Heterogeneity, I just described the variance in the different sizes. And then these distributions. And so, I'm kind of going to lean off the distributions, but that just kind of shows us, at a glimpse, if it's an older forest that has a lot of different ages and different sizes, or if it's a young forest maybe, and they're all the same size and they're just kind of coming up. So I'm going to jump right into some results. First of all, we see that there are indeed positive relations. So this is a good sign. The strength of the relationship is not super strong. If it was very strong, you would see this on a one-to-one line. But nonetheless, they're significant. And so we know that we're kind of on the right track. So if we're to take the same sort of analysis, this is just an example with 4 of them. But now we have all the different pairwise combinations of diversity and structural metrics. And some things start to pop out. The first is that stem density is clearly related, so just the number of trees. And so that's partly explainable by things like resource availability. So maybe if we have a fertile soil, we would expect more individuals and at the same time, maybe more species by potentially sampling effect. In other words, just the more that there are, a random draw, you would just expect there to be a different species that would pop up. If there's only one individual there, you're not going to have 2 species. But if you have 100 individuals, you might expect to see several more species. Just as a sampling effect, nothing really about the underlying ecology. Maximum is another thing that comes out. And you can really see that these Spearman relations, where 0 is no relation, and 1 would be a perfect relationship. These are actually quite high for this sort of ecological study. And a couple explanations, which I propose here, is a site index. This is a forestry term, which just kind of says like how fertile the site is, not just the soil, but other conditions. And so again, with that sort of inclusive or benign environment, then you might expect there to be more species that are able to colonize and find a niche, recruit, and take hold. The next could be time since stand replacing disturbance. In other words, how old are they? So if you see a forest with really tall trees, there's a good chance that that's an older forest because it takes time to grow tall. Now, of course, you have individuals that grow faster than others, a lot of confounding factors. And so this time since disturbance, or the age of the forest, also provides time for other species to come in and colonize by stochastic or chance events or by other deterministic events. This is agnostic as to how they come in, but we do see that relationship. And finally, heterogeneity. And this is what I get excited about, structural diversity or structural heterogeneity. And so what that's saying is, for example, with this CVBA, that's the coefficient of variation of basal area. We talked about that before. That's just the variance in different sizes, the girth of the tree. Well, you could say the same thing about height. You have short trees, medium-sized trees, tall trees. This seems to be highly correlated with all of these measures of biodiversity. And there's a few customary explanations for that. One of those is niche complementarity. Don't want to get too into the weeds on the ecology here, but essentially what that's saying is that the more that you have a structurally complex canopy, the more chances you have for light angles to hit the ground in different places at different parts of the day or different parts of the season, which may or may not help certain species establish. The same thing could be with root competition for water and nutrients. The more spatial complexity there is, the more chances there are for different species to exploit their traits to be able to establish and potentially outcompete other species. And so that gets into spatial resource distributions. It's kind of the same thing. The final thing I'll just say, morphological traits. And this is a little bit of a chicken and egg here, where the more, let's just take it from diversity. The higher the diversity, without knowing the identity of those species, the more size diversity you might expect. Because if there's more trees, you might expect expect some are going to be short, some are going to be tall, some are going to have a conical shaped crown, some are going to have a spherical crown. All of these different things, when they combine together, are going to create a complex canopy. And so just based on that alone, that you would expect some sort of correspondence between structural diversity and taxonomic or species diversity. So a final thing here, and I'm not a huge fan of this sort of measure in R squared, but I said, okay, let's just throw everything but the kitchen sink at this. In other words, let's throw all of these structural indicators. Nothing else, nothing about climate, nothing about soils, just structure. And if we put them all together in a non-parametric model, in this case, support vector regression, how much of the variance and diversity does it explain? And we find some pretty good numbers here. Between 44 and 60% of these main kind of full categories, it means all plots. And in some of these individual subsets, we see that it's explaining up to 71%, specifically in these artificial, these plantations that have been manipulated for forestry purposes. So this is all encouraging. So just to kind of wrap up on this first part. So we do in fact see significant relationships in this area of the Piedmont between structure and diversity. And we're really seeing it with density is a huge one with sampling effects. Height keeps coming up as a predictor of species diversity and heterogeneity. Put these together, models can explain up to 70% of that variance without accounting for all of those other things that ecologists consider important, like water and nutrients and climate. And then the next question, and this is where we're going, is how can we use these empirical field findings to inform some sort of predictive modeling using remote sensing data? Idea being, if we can remotely sense structure, can we use this relationship to remotely sense biodiversity, which is something that remote sensors can't really do, maybe with a few exceptions with hyperspectral and some of those things. But is this a way to have a broad scale mapping model of biodiversity? Okay, so that's where we're going. So the next step here was to kind of, baby steps to do this at the landscape scale. I wasn't going to start with a global model. I was going to start at my local landscape. And the 1, 2, 3 of this, and it's what I just kind of outlined, is that you start with this field data, robust field data on what species are there, what's the sizes. Then you can use an airborne LiDAR system, which enables you to characterize the 3-dimensional volume of those forests. So now we have our structural component. Wrap this up in a model, a little bit of voodoo and some magic thrown in there, and you can use these to predict the distribution of diversity, and importantly, predict the certainty. And so that's a very important thing with any sort of mapping project, is not just to predict what you're trying to predict, but also to be able to have an associated layer of uncertainty, a spatial layer of uncertainty in those predictions. So, okay, so 1, 2, 3. Here's number 1. Field plots. Had an awesome crew of undergrads, some master's students with us going out in the field using this nested sampling design. So essentially what we're doing here is we have 30 by 30 meters. It's nice, kind of corresponds to a Landsat pixel, classic optical satellite, and break that up into 9 different modules. And within each module, we do this nested subsampling. And so we get smaller and smaller, all the way down to 0.01 meters squared. You multiply it or you square this, and it actually becomes a smaller number. And so we can't do this sort of sampling at the whole extent. It would just take forever. So we're actually sampling, we take all of these, and we're hoping that these nested random corners are going to be indicative of the biodiversity in general across scales. So this is kind of a cool part of it. And because it's a geography department, I'll quickly get into this, but then, and if anyone's interested in this with field sampling, let's talk about this later. You go out there in the field, you have a GPS with an antenna, it's kind of fancy. What's the accuracy, estimated accuracy? There's a sigma of about 10 meters, okay? And if you're trying to map using this high resolution LiDAR, and your accuracy here is 10 meters, I mean, that's kind of garbage. You can't work with that. So what we did was we got this amazing orthorectified high resolution imagery here. And we looked for interesting things, like one of my favorites is if you look at like some of these places, I think maybe this is an example, where you can see a tree, you can see a tree stem, and then you see its shadow. And where those intersect is the ground, right? And this is orthorectified, which means it is fairly accurate at being, this is the precise place on Earth, geolocation on Earth where these elements are. So in the field, we actually took out measuring tape. We found all this. Sometimes it would be a rock we could see in the imagery. So we take a measuring tape to the rock and we go to the vertex of the plot, and then we do another and another. And so you can triangulate. And so what we could do is we could go into GIS and say, now because we just triangulated the positions of these vertices, we know with close to sub-meter accuracy where those plots are. So we threw the GPS away and we did it all in a GIS. Well, we didn't actually throw it away. We just put it in the closet. Okay, so airborne LiDAR, NASA G-Lite, they've flown all around the country, done a lot of stuff in Alaska. They flew this tract in North Carolina. How many people here are familiar with LiDAR data? Okay, all right, so we have, looks like about 70% of 50%. So you do the math on that. I don't know how that works out. So essentially what's happening, the quick and dirty of this is that, well, let's go back. All right, we've got a plane. We have this LiDAR sensor. It emits a laser pulse, which then bounces back to the sensor. The laser goes at the speed of light. So if you measure the amount of time that it took to go down and back to the plane, we can calculate this distance. If this is the ground surface, then we know how high the plane is off the ground, and we can use GPS to calculate what the elevation of the ground is. If that's a forest canopy, what you may see instead is that it's hitting the top of the canopy here, in the middle of the canopy here, and the bottom of the canopy here. So we can start to use that to create these little points in 3 dimensions to see what's going on with the 3-dimensional distribution of the canopy. We call this a point cloud, right? And from that cloud, we can start to make these really intricate, hyper accurate, precise models of forest structure. You can see some Doug firs over there. You can also see this kind of open woodland landscape over here. I think it really helps for that, to see how it's capturing that structure. Okay, so we can put these in a GIS. We have these derived layers. You can see a subset of this. This is a canopy height model, which is just saying how tall is the canopy. Here's a little subset of that, and you can see that detail. It's really capturing. By the way, this is the location of the FACE experiments, major NSF experiment. They cut this out and they did CO2 fertilization to see what that did. So this is the study area on the upper right there. Some weird stuff going on there in Duke Forests, clear cuts and stuff. So I kind of cut those out. That wasn't really interesting for me. So we end up with this kind of awkward shape here, but that shape does correspond to relatively natural forests. So what are we getting out of this? Okay, well, again, lots of derived layers, derived indices. I'm not going to get into it, but I'll just say topography is a big one, the last return. In other words, that shot that goes all the way down to the bottom and all the way back to the top, that's the last one that comes back. That's the one at the ground. Tells us about the topography. The first return is the first one that comes back because it hit the top of the canopy, and we know how tall the canopy is from that. And then everything in between tells us about that structure, everything in between the top and the bottom. Okay, that's strange. There might be some Mac PC conversion issues here, but this is cool. So one of the things here, I talked about doing this like fine scale diversity, right? Or measuring fine scale diversity, and we're going to be mapping that. And that diversity is actually often understory. It's underneath what the LiDAR can see. It might be obscured from leaves that are above it. Subpixel, it might be that you have one pixel of the LiDAR and you have tons of different species there, or you only have one. We don't really know. It's intertwined with neighboring taxa. There could be two different kind of species growing together. And so how do you do this when you can't see it? Well, honestly, modeling approaches, because there's no way, kind of physical based way to get at it. So what we did then was we took this LiDAR data. This is a TWI, is an indicator of soil water. And we aggregated at these different spatial scales. And then we can ask these certain questions. How are these predictors doing at predicting species richness, SR, which is on the y-axis, at these different scales, spatial scales, as we go up? Well, the first thing that we see is kind of a definite pattern of scale dependence, right? We see that it's improving, generally getting more positive as you go to these larger spatial scales. And that's partly due to a few of these issues of imprecision. The larger you go, the more you can kind of assume some of that imprecision. And then characteristic scales of biota. So some of these are big trees, right? So if you're measuring like really small down here, that small scale biodiversity is not going to help you if you have a huge oak tree. We also see that topography ends up being a big factor. In this case, the taller you are, the drier it is, and the fewer species you see, the lower you are. You might be in that river bottom where you're going to see a lot of those small species and a higher species richness total. We're also seeing that these kind of mid-structural indices are doing really good, and they're generally positive. The more stuff there is in that mid canopy, the more species we're seeing. And that's the same thing we found in that prior study. And then finally, and this is the bonus, I didn't do anything on hyperspectral, and there's a big part of this paper and other papers where I'm also looking at the diversity of the hyperspectral signal. And this is a talk about LiDAR, but this is just a bonus. And what it says is it's not great, but spectral variation of the hyperspectral signal actually ends up being a really good indicator of biodiversity. A lot of explanations for that, but again, this is just a bonus. This talk is not on that, so we're going to move on. But again, you can think of diversity begets diversity. Diversity of spectral signal correlates with diversity of taxonomy, which makes sense because all the different species have different biochemical characteristics. All right, so once again, we have these different variables here. We throw all the variables kitchen sink approach here into a random forest regression. I instituted this feature selection and bias correction routines to be able to really hone in on those variables that were most important. Sometimes if you throw too many variables, it actually introduces noise. So you try to select the perfect amount for each model at each spatial scale. And then you make a relationship, and then you flip that relationship around and you use that for projection or for prediction, excuse me. So here's what a prediction looks like. So here you recognize this space. And what we're doing is we're mapping the different scales of biodiversity. So here it's actually saying what is the expected diversity, and I should have said this earlier. We're talking about plants here. Vascular plants. So how many plants would you expect to find in this little 0.01 meters squared? That's like this, that's pretty small. And a lot of it is zero because it's just going to be leaves on the ground, especially up here on that hill. You can see over there, we've got a hydrological layer, and you can see how the mid right part is kind of going up this hill. It's quite dry there. Well, there's not as many plants. And then once you get up to the top, you see that there's a little stream, and that jumps up to 4. So that's kind of showing that there is, based on our field data, there were a lot of small species that were all crowded together. Once you start to kind of degrade the signal, but but still based on those models, but instead of the 900 meter square, which is the largest square, you see large scale, you see some of the same patterns, but some noticeable differences. And to really get into those differences, you go back to what I just showed you, those plots where you're doing inference on every single relationship at every single scale. But this is a visual indicator. And it's also kind of an example of how this can be used to understand prediction of diversity across scales, because especially when you're doing something like species richness, the number of species, scale matters. The larger you are, the larger the area, the more species you're going to find, just because there's more room for them. And so you need to figure out ways to control for that. So as I said before, prediction is important, but also associated with uncertainty. And so we used a random forest, which is a non-parametric predictive algorithm, to produce the diversity patterns here. And at the same time, what it does is it spits out a posterior distribution. So it shows us kind of all of the different, let's say, possibilities for what an individual pixel value could be. And so if there is hypothesized, in other words, if there is a lot of agreement about what that prediction is, you're going to see that the distribution of what it thinks is very narrow. So it's very confident that it's a certain value. Whereas if that model is not really working that good at that scale or based on those predictors, it's going to be a lot more broad. So there's going to be a lot more variance. And so that's a kind of backhanded way to get at our certainty, right? Because if we're really peaked and we're very certain, if we're kind of broad, it could be all sorts of values, but we kind of think it's this median or modal value, which is what goes into the prediction, then it's a low uncertainty. So these are two totally different maps. This is the prediction. This is the C. It's a coefficient of variation. It's that spread, that variance in the prediction. And so what you can do with this is you can play with color combinations to be able to map these on top of each other. So you can see this kind of space from high diversity, high certainty to low diversity, high certainty, all the way here, low, low, high, low. Kind of mix these up. And this gives us a kind of idea, not just of the diversity distribution on the landscape, but also how certain we are in the prediction of it. So just to conclude, this research demonstrated the capability of mapping landscape community distribution, and specifically biodiversity distribution, using an airborne sensor from NASA G-Lite. And the predictive maps are inclusive of things that you can't actually see with the remote sensor, which is subpixel understory taxa. We're relying on models for that. And that's going to add to our uncertainty. It's true, but we're going to quantify that uncertainty so people kind of know that caveat emptor before they use the data. I'll just plug really quick a kind of sister paper of this was the same site. And instead of mapping diversity, I mapped community composition. And specifically in the context of community continuum, which is this idea that in fact, there are no two communities that are alike on the face of the earth. And we often say things like, oh, that's an oak forest, or that's a pine forest. Well, when indeed, there's probably some other stuff with the pine. There's probably some other stuff with the oak. Everything is idiosyncratic and new. And so how do you think about that when you take a mapping approach? And just very quickly, because this isn't a talk about this, here you can see mapping with two basic communities. I use clustering algorithms to figure out the most parsimonious way to split these into two. And you can see that kind of up slope coming in to create this upland sub-xeric. Well, let's split this into 7 communities. And these are this kind of compositional space, these different communities. Okay, so then we're starting to partition out now into finer and finer resolution. So this is 2 communities, this is 7 communities. This is a continuum where I use this kind of red, green, blue combination to represent this continuous space of compositional variation, which is indeed how we experience it when we go out in the field. So anyway, this is an example of an application. The research highlights the role of topography. So, I mean, maybe that's kind of a no-brainer. The closer you are to the river, you're going to find probably smaller plants. And if you're in a dry area, there's going to be fewer of them up in the dry spots. Structural complexity, like we found in the last section, really important for predicting the community dynamics, like all these things, competition, recruitment, all these different things kind of get wrapped up in this structural complexity, which can help us predict species diversity. And then finally, biochemical diversity, which is the spectral entropy. In other words, that's the hyperspectral signal. But that was like a really big part of the paper. There was a lot of a high predictive power was just coming from that hyperspectral signal. Finally, this is a cross-scale analysis. Scale is a big part of it. Scale in the field sampling, scaling in the LiDAR and the modeling. And we found there was indeed a signal for scale dependence, which, like I said, subsumes this imprecision. So the larger the scale, the better the models. And it aligns with characteristic scales of the biota. And there are going to be other systems where these scales will not hold. For example, in a grassland. In a grassland, you're going to start to see the power saturating at a certain point because the individuals are just much smaller. And so that would be the characteristic scale to model them. Okay, so moving on to number 3. This is the biogeography component. This is just out. It says it was accepted in July, but it took them a while to prove. So this just came out like a week ago. It's an early release. Go check it out. Yeah, this is representing some recent work that I've been doing. And so here again, we're thinking about relationship between taxonomic diversity and structural diversity. But instead, I have been focusing on this landscapes, and there's other studies, someone in Czech and someone in Colombia and, different places on the earth, and they're finding these relationships, but, how do they generalize at much larger scales? And what's underlying that, how they play out over those scales? And I say, or I, propose that it could be climate is a major factor. And so another way to kind of think about it, and the biogeographers like to think about this interconnection or interaction between region, which is climate and other larger scale factors, and local effects, which here I'm defining as forest structure. And thank you for not catching that spell check. So, but you know what it says. All right, so I'm using the NEON data. How many here are familiar with NEON, NSF NEON? Okay, cool. So now we're at like 40%, but everyone was like 100%. So that I can deal with those statistics. This is the way that NEON splits up the country. They have sites, terrestrial and aquatic. I'm dealing with terrestrial, that have a consistent sampling design where they go out in the field and they measure all sorts of stuff. They got flex towers. They've got a plane that goes over and does that sort of airborne LiDAR that we were looking at before. This is how they split up the US into these discrete areas. Well, if you didn't already know, with my last little spiel on continuous and continuum, I see this and I kind of bristle. To me, they're kind of like state borders. Like, are you telling me that this thing stops there and this thing picks up right there? So I instead thought, okay, well, let's think about this in terms of these reduced dimensional spaces. And unfortunately, it's not picking up all these colors. It's coming across as white. But this is indeed, this should show up as light yellow. In any case.
They can see colors better on this.
Perfect. Okay, so that's for you all then. No. So what this is kind of showing is that climate is indeed continuously varying, and it's varying in different axes. So you can have stress here as aridity. You have this whole gradient of aridity, but then at the same time you have a gradient of energy. So you could have high energy, which here is mean annual temperature, and which is orthogonal or totally different, perpendicular to that stress gradient. And then here's another one I threw in because I love RBG, right? Red, green, and blue. RGB. I think I said RBG, which is because Matt has this funny mug that I used this morning, which is about Ruth Bader Ginsburg in those three colors. So clearly I was going to get that wrong later in the day. But anyway, the third one here is seasonality, precipitation seasonality. And so here are the neon plots that I'm using, and it's a way to see how these plots differ in this continuous environmental space rather than those discrete categories. And so it's going to be a way for us also to characterize why we might see differences in structural biodiversity relationships in Washington State or Alaska versus Puerto Rico or Maine. So with climate, just going to run through this pretty quick. We've got the stress, coldness. How cold is it, right? Below zero, preferably. Water deficit, right? Aridity. For energy, what's the temperature? What's the actual evapotranspiration? Matt's going to have to explain what that is later. Seasonality. Temperature seasonality, precipitation seasonality. How much do we see variance across the year? Is it raining equal amounts, or do we have a pronounced rainy season and dry season? So then from these sites, we can kind of pick out the LiDAR, the structure, and the diversity from the different plots that they have in each of these different sites. So here quickly is an example of some of these plots. Does this look familiar to anybody? Yeah, we just saw that, right? Okay, so my people in North Carolina were actually the ones that were able to convince NEON, this national program, to adopt their sampling scheme. So I love it because I hit the ground running. I'm like, NEON, I've been working with that for years in North Carolina. In fact, I even did my dissertation on a variant of that, right? So we have multi-scale biodiversity. We have some of the same things, these nested corners. Structure, well, I thought about it in terms of three different variables, and or let's say three different axes: dimensions, heterogeneity, configuration. In each of these three, try to bring in a horizontal and a vertical component. So with dimensions, well, height, well, we got a plot, but let's call it cover. So in other words, how much of that horizontal space is covered with trees? Structural heterogeneity, vertical, how much, like, how crazy is it in the vertical dimension? How heterogeneous is it in the horizontal space? Spatial configuration. Configuration is a little bit tricky. So here we do clumping and skew. So clumping, you can see in that bottom right, heavily clumped canopies, which means a little here, a little over there, space in between, versus continuously dispersed. Skew, well, that's our vertical measure. And what that's saying is, are we seeing forests that are very bottom heavy with lots of biomass here and then maybe a few kind of scrawny things sticking up? Or are we seeing something that's like a pine forest? It's really tall with a thick canopy and then nothing underneath, so it's top heavy. Or are we seeing something in between? So I've been through a lot of structural metrics. I've done a lot of exploratory analysis on this. And I find these sticks are really parsimonious and they kind of cover the space of structure. And so I use this as this kind of simplification. There's a ton more out there, but these are the 6 that I'm using today. So with LiDAR, the next question is, well, we have this 20-meter plot, 20 by 20 meter plot. How much LiDAR do we use? Well, if you can imagine that this plot, we measured biodiversity on the ground. And if we're saying that structure impacts biodiversity by kind of altering the light and nutrient environment, well, we know that over the course of the day, the sun angle is going to change. And we know over the course of the year, seasonal sun angle is changing. So just right there, we're starting to think, well, maybe we're having the sun coming in at this angle. So maybe actually what's happening outside of the plot might be more important for what's happening inside of the plot. So whenever you kind of speculate on these things, well, you got to test it. So I'm glad this didn't crash. Here are 2 different approaches. One are concentric rings. So these 10-meter rings. How does the explanatory power of this structure change as we go further and further out? Explanatory power explaining the biodiversity inside the plot. Or do we use an accumulative circle? But here, we're not actually, neither of these are we controlling for area. So we have to do that statistically. But this is getting larger and larger. And how big do we get before we get too big? And then it's actually not explaining diversity of the plot very well. Because now we're outside of the root zone. Because remember, you can have a tree established here that has roots coming all the way out here that's competing. And then also I explained the life dynamics. So what is that scale? Well, here's some of these kind of across different ecoregions. I'm just going to jump right to the chase. And what we see is that we see this pronounced peak larger than the size of the plot. And I actually use those concentric circles, and I found something fascinating. Structure 100% outside of the plot predicts diversity better than structure 100% inside of the plot. So one thing is totally unrelated to the other, because that structure outside of the plot is the thing that's actually controlling the light environment inside of the plot. So that was kind of cool. Not something I expected, kind of fascinating. In the end, we decided to stick with the circle approach, so not a ring. So it includes the plot. It also subsumes some of the area outside of the plot, 2,500 square meters. Okay, so a lot of stuff here, but this is kind of like that slide we showed before with the Spearman correlations. These are individual predictors of structure, individual response of diversity, and these different colors correspond to those sites and plots in that three-dimensional climate space. I don't have this labeled because I don't want people to think about those labels. It's not about what site is where. It's really about, oh, well, this is a site with high energy, and this looks like a cold site over there with seasonality. So you can start to see these patterns unfold. The quick, thing to take away from this is that we're seeing quite high correlations across all these sites in the US, even though some are negative and some are positive for tree diversity, SR against species richness. Don't worry about H, that's another one of those diversity indices. And we're seeing lower relationships, or less strong relationships for plants. Well, that's like a little bit too crazy for me. I want to break things down, use Bayesian analysis, multivariate hierarchical analysis to really do inference, to look at the effect that these different components are having on diversity. So the first question is, well, how does climate drive diversity? Well, we see a few things. First of all, we see that coldness has a negative effect, right? Anything here is negative. Anything that crosses is not significant. Anything on that side with the filled in is a positive effect. So we see that coldness, the colder it is, the more coldness, in other words, the colder it is, the less diversity you see. Kind of, I'm gonna say maybe that makes sense. Aridity, turns out, doesn't have an effect, which is a little surprising. And then I remembered, hey, South African finbows, the coast of North Carolina, these are places that are actually very arid and have tons of plants, tons of biodiversity. And there's other places in the US that get tons of rain. They don't have a high biodiversity, like Oregon. I mean, there's places in Oregon that have, don't get me wrong, I love it, beautiful Doug firs, but there's not the tons of different plant life, flowers that you would see in the North Carolina coast. So that lends partial support to this physiological tolerance hypothesis, which essentially says that the more stressful it is, the fewer species are going to be able to deal with it. The next is energy. So evapotranspiration, actual evapotranspiration, right? How much of that water is cycling? Temperature, totally positively related across the board. The more temperature, the more climatic energy, the more species there is. And that supports this species energy hypothesis, which kind of explains itself. More energy, more species. Biogeographers have been studying this for a long time, which helps explain the latitudinal gradient in diversity, where we see lots of diversity towards the equator. Those diversity levels start to taper off closer we get to the poles. Next, results inconclusive, but generally speaking negative for seasonality, which supports another hypothesis, is the climatic stability diversity hypothesis. So stability is the flip side of seasonality. But what the stability diversity is saying, the more climatically stable it is, the more time plants have to grow, produce biomass, compete, do everything that we think of when we think of these high diversity places. So the more seasonality there is, it's the opposite of stability, the less diversity we expect. And we're generally seeing that, although they're not significant in several of the cases. So it's a good indicator, but it's not significant at this level. So we'll drop it for now. Okay, the next one here is how does climate drive forest structure? So we did climate on diversity, now climate on structure. Two quick takeaways. Water availability is huge. All of these climate indicators associated with water are significant. And they might be negative, they might be positive, but they're significant, right? And maybe they're negative or positive, just the way that we define it, but they're drivers. Temperature, on the other hand, oh, see, lots of need. Not significant. Every single one is not significant, okay? Climate driving structure. So in New Hampshire, big bushy forests, cold. Florida, big bushy forests, hot. Same thing. Temperature, not a huge indicator of forest structure. Okay, but this is the meat of it. How do they interact? Okay, there's a lot going on here. It's all here. So I'm just going to try to break out a few of these different things, okay? These are partial effects, which means we separate everything. We separate the role of structure, climate, that's the interaction over there. So just really quick, plant diversity is negatively correlated with cover and height. So the more that we have openings, the more plants come in. Positively related to horizontally heterogeneous, clumpy, bottom skewed canopies, right? So plants like that, bottom skewed, we might be just actually measuring the plants that are down there, the lower story biomass that's there. Climate corroborates the previous findings. We just went through that, coldness, not aridity, energy, stability. Okay, but interactions. So this is kind of where we're going here. So there could be a lot of discussion of this, and I'm going to go through it kind of quickly. But we see an inhibition of plant diversity under taller canopies. We see that inhibition actually decrease the more cold it gets, and increase with the energy, like with temperature. And this is, I suggest, I speculate, hypothesize that this is an effect of microclimatic buffering. In other words, the more tree, the more canopy there is, the more it protects that understory microclimate from the stress of the outside macroclimate. I'd love to talk more of this, and let's talk later, but I'm going to move on. We see some of the same thing with tree diversity, except in this case, tree is positively correlated with cover. So the more stuff there is, the more tree diversity we see. Vertical heterogeneity, okay, that's what we expected. Climate corroborates what we saw before. Again, these same patterns, we would expect that because we just found that. In here, we see that the role of structural heterogeneity increases with climate energy. So the more energy, the more temperature. We're actually seeing the structural heterogeneity is an even stronger predictor of diversity. Why is that? Well, you really need to do like a mechanistic experiment to get down to the bottom of this. I'm kind of doing data mining here. But could it be morphological diversity? Once you start to get to those warmer climates, you have different shapes, different crown sizes of the different trees. Could it be asymmetric competition, which is essentially saying that there's different levels of competition based on access to light at different parts of the canopy in these more southerly, lower latitude forests? Remains to be seen. This is a really exciting research topic. So just to conclude here, we have evidence of these continental scale biodiversity structure relationships, stronger for trees than plants. We see climate concurrently explaining regional variation, diversity, and structure, where it's energy, stress, stability affecting diversity, and water availability, but not importantly, not temperature, that is exerting effects on structure. Finally, the two forces, climate, regional, structure, local, interact to mediate diversity along these primary gradients. That's what this data showed. There may be more, maybe other data disproves this, but this shows coldness, mean temperature, and AET, evapotranspiration, actual evapotranspiration gradients are the ones that are most strongly interacting. So I'll wrap up now in a couple of minutes and just briefly kind of talk about what I and my lab is up to in terms of forward-looking research, emerging applications for this sort of thing. So first of these is we are members of the JEDI science team, and my mentor is the PI of science, deputy PI of the project. This is a spaceborne LiDAR on the International Space Station there. It's on the Japanese arm, and you can see that there is the JEDI sensor is located right there. So it's going around the Earth on the International Space Station, and it's doing the same thing that the G-Light plane is doing, except over vastly greater distances. And the data product, it ends up being very different as well. Here we can kind of see just a quick snapshot of how that looks. So in A here, you can see we had an airborne LiDAR run over this area of Santa Cruz. This is actually what ended up burning up in the CZU fire. These are redwoods. And you can see that airborne LiDAR does a great job of capturing the forest structure across the landscape. And you can see this profile of this redwood tree going all the way up to 80 meters, huge tree, okay? And here's JEDI that comes in, because it's coming in from space, it can't do everything. And it has these individual pulses that sample a footprint on the ground. And there's distance between these footprints in the along track direction, and there's even greater distance in the across track. And so it becomes a question of creative remote sensing, as well as innovative statistics to be able to interpret this, to use this sampling design that's inconsistent, or it's consistent, but it is not spatially continuous across the landscape. So we have tons of these tracks, they're piling up, the data's coming in. We just, is a major accomplishment. We managed to change the altitude of the International Space Station because we were locked in this orbital resonance, and the Russians wanted it at one altitude and we wanted it another, and it was head to head, and then they moved it. Yes, we did it. And so hopefully that's going to help us to improve these gaps here in between the orbits to get more and more data. And we just got extended for 2 more years. So it's been up there for 2 years. We've got 2 more coming up. And so again, this question, here's with a neon site. You can see these different neon plots. Neon has LiDAR for all of this, but then how do we interpret these JEDI points that aren't everywhere across the landscape, but they are everywhere across the globe, except for the poles. It's at over 50 degrees. And so this is kind of what's consuming our lab. And we have the supercomputer running, and we're doing cloud computing, and we're doing lots of stuff. We got ecoinformatics grad students working on this to try to be able to reconcile these things. So we can do inference and we can do modeling at the global scale and not be stuck with these little planes that can do one little small extent LiDAR. Does anybody know where this is? A little geography quiz. This is a tough one. Here you can see that this is Sentinel imagery, and you can see that there's some serious issues with the bidirectional reflectance distribution function here, which is kind of showing this these artifacts here. This is the island of Borneo, split between Indonesia and Malaysia. And this is another project we're working on, where we're working with landscape ecologists. We're doing species distribution modeling of some of these cool species. And we're looking at using the LiDAR from JEDI and these optical instruments and radar, and then all this amazing camera trap data from Oxford to be able to see where are these critters, where are they going, where is the hunting pressure, and how can we optimally plan and manage conservation schemes in Borneo and in the mainland, which is Southeast Asia, and in other places in Indonesia, to be able to maximize their chance of survival. And so one among many applications of JEDI LiDAR, because all of these, they respond to structure. If there is a lot of structure, some of them might really like that. Some of them might not like structure. They might prefer the wood, more open structure, still structure, but it's an open structure. And so if we know their habitat proclivities, we can model that, and we're using circuitscape and other types of connectivity analysis to see where they're going based on this camera trap data. So I've taken about an hour, which was as much as anyone probably wants for any one day. So I'm going to wrap it up and just say I've talked about my own research, but there's a ton of research on this, and go check it out. And this is a great old school study from 2005 on understanding complexity and diversity and structural diversity. We have a paper coming out, a special issue in Frontiers in Ecology and Environment, with all these cool papers. This is happening. Hopefully this will come out in about 6 or 8 months if everything goes well. Keep an eye out for that. And thank you to everyone who's made this possible. Some major collaborators. One of them is in the room, Dr. Denberg here. All these other great folks, funding agencies. Chat me up after the talk. Come out to Flagstaff. Let's take the dogs for a walk and we can talk science. Thank you.
All right, we have any questions for, break my computer. Zoom people, we can hear you, so feel free to unmute yourself. Oh man, Friday afternoon. Yes, we hear you.
So the benchmark diversity thing seemed to be species all the time, or some metric of species diversity that's rated by something. But it seems like with all the new tools, it's possible that diversity and biodiversity might start to mean something else. That instead of like using structural diversity to predict biodiversity, which means species diversity, biodiversity might come to mean structural diversity.
Uh-oh.
What do we do?
What happened? It looks like the Zoom link went away.
Well, Susan's still not. Yeah,
It's just the visitors are up here.
yeah.
Hey, George.
Hi, hello. How are you? Looks like we are cut off though.
We're in exile.
Yeah.
We have the exile crowd.
I read that, but same time. time
Now they're back,
is
maybe.
one of the oldest metrics, and it's something that's easily relatable. And so that's why I chose to focus on this for this talk. And my research is indeed employing those other alternative metrics of species diversity or biodiversity, as I say, to look at these relationships. And yes, and also structural diversity is a part of biodiversity, and it's a shorthand. I say biodiversity when I mean taxonomy. Cool. Hey, everybody.
Yeah, Zoom stopped working here,
Okay.
so we're now using the
Oh,
computer.
we're back.
Sure.
Can everyone hear him now? Except now we're not going to be able to hear. Well, okay, they can hear you, but we might or might not be able to hear them. Do we have a question on Zoom?
I'll try if you can hear me.
Yeah.
Yeah, I can hear you.
Okay, good. So you've got lots of significant relationships for lots of different variables related to lots of different hypotheses. Are you looking at a way to narrow this down to essentially to see? And the big answer is yes, it's complicated, but can you uncomplicate it in some way? Are you looking at a way to at least rank the relationships or the hypotheses that you're supporting?
Sure, yeah, great question. I would just answer it and just say that two totally separate endeavors. And one endeavor is prediction. So that's optimization of predictive models. And so there, we may not even care about hypotheses. We may not care about variables. All we want to do is maximize our predictive accuracy, which can be very helpful for conservation planners or policymakers to be able to map diversity or structure and then be able to make actionable decisions based on that. The second avenue kind of gets to the kind of core of ecology, which is getting into mechanism, which is something that clearly I've been alluding to, which is another important thing. And that is what's driving this relationship? What's the theoretical underpinning and what are the implications for our understanding of forest ecology? And there, I would say that there are a few kind of prominent hypotheses that can be assessed either through experimental manipulation or through some sort of large-scale data mining. And those are essentially, they refer to the basic community assembly processes. How do species, how do individuals form communities? And so I mentioned one, which is niche complementarity. So the extent to which different, in the spatial heterogeneity in the environment corresponds to different niches for different species. Things like selection effects, which is, are there certain species that have functional traits that allow them to have a different structure or have a different functional outcome on the community? A sampling effect, which I talked about, which is statistical. And so I think those are three of the dominant ones. Mass ratio effect is kind of related to the second one that I said, the selection effect, which is essentially there are certain individuals that have a disproportionate effect on the environment. And so those are the sorts of things that I think that the hypotheses, the basic hypotheses you can use to frame and kind of simplify this kind of dizzying look at with so many different variables producing so many different relationships to kind of to boil that down.
Okay, but can you rank those? You also had mentioned like climatic stability, diversity. Can you get an idea of their relative importance? They're all explaining part of the same thing, right? But and I expect that some are more important in one place than in another, and there may even be a biogeography to explanation. But that's what I'm kind of interested in, is how you look at the relative strength of an explanation in different places.
Yeah, entirely. And so this is not something that I reported on here, but with all of these, for example, bivariate relationships, you can get explanatory power out of that. I didn't report any of this, but Temperature is a huge driver, often outweighs any local effect, including hydrology, soil, and structure. And then there are several cases where having the instrument, the LiDAR instrument, shows that there's structure there. And you could even envision it as a woodland where you have trees and then you have a big space in between. If you didn't have an insight into that structure, you might predict that there's a constant tree diversity, but indeed with the LiDAR, we can just see, we've actually just measured it. It's not even about modeling it. We just see the tree here and we don't see it somewhere else. So that would be something that becomes much more important, let's say in a heterogeneous landscape, whereas a homogeneous landscape, it tends to be some of these larger scale factors, which could be dispersal in the landscape, or it could be climate at a much larger scale.
Okay, thanks.
Sure. Yeah.
So I was kind of, this is something that you mentioned a little bit in both sections 2 and 3, which is like kind of the scale dependence question, like what happens as we like aggregate to larger and larger scales to like predictive power. And in section 2 of the talk in particular, you showed that kind of like as you increase the spatial scale, predictive power gets a little bit better. Is that right?
Right.
And I was wondering, and I think you sort of like addressed this in section 3, but like, is there a point at which that starts to saturate or even switch to the other direction, where continuing to aggregate, you're getting to a point where now you're beyond the relevant plant scale and the predictive power actually goes down? And if so, does that have like negative implications for trying to scale up to like a global scale, where you have to kind of start aggregating over larger regions? Does that make sense? Like,
yeah,
I guess I'm kind of getting at like,
it does.
Like, what are the challenges associated with like a global scale mapping, given that potential saturation?
I see. Okay, I'm just going to take those as 2 questions.
Yeah, sure, please.
And the first one, I'll just point out that like with scale, we're talking about resolution and extent. And so I'm really talking about increasing resolution. And so you could measure at a global scale, and that could be irrespective of the resolution that you're modeling at. So I'm thinking of it in terms of resolution.
Sure.
And so that resolution, you know, as I said, it's part of it is imprecision, but a big part of it is characteristic scale of these communities. And so you're totally right. As you start to get larger and larger, you're no longer at the characteristic scale of those individuals. And you can start to get idiosyncrasies of the landscape, which could be an open patch, it could be gaps, it could be a thick pine forest or something like that. So that at that larger scale, you certainly do see a saturation, you see a drop off. In section 3, I talked about that with the LiDAR, which is the same thing across all these ecoregions. You see a peak actually larger than the plot, but then it starts to go down because you're adding in noise. It's kind of saturated out. To take the second part of your question, if I can paraphrase, which is kind of, you know, what are the challenges to instituting this globally? So there's many of them. I think a big thing is data. Scientific data, ecological data is tremendously biased. We have tons of data from North America. And it's NEON, kind of, I wish NEON's, this consistent sampling design was spread around the world. It's not. So we don't have a lot of good data from the tropics. So that's going to prevent us from modeling the tropics. If we're relying on this LiDAR, which is dependent on cloud cover, we have cloud cover, it's not going to work well. It's the same with optical imagery. You have that again in the tropics, you tend to have tons of clouds. That's giving us less remote sensing data from those areas. Okay, so those are two things. And so then you start to get into the problems that I kind of elaborated on with JEDI, where it's a sampling scheme. So we have global coverage, but how do we characterize what's happening in between? And so that becomes a modeling exercise, it becomes a computer science exercise, because what you can do is you can do some sort of fusion, let's say with an optical imagery, which is spatially continuous, but then that's introducing its own biases, because now we're just basing things based on optical imagery. We're making some sort of interpolation, we're making a prediction about what the structure is there, even though we don't have JEDI. And so that ends up being a whole other problem. And so we have to do the same thing that we did in the second part, which is we need to do prediction, but we also have to model uncertainty to know where we're getting things right, where we're getting, or where we don't know how well we're doing, right? And so I see all of those things as being primary limitations. On the plus side, there's more satellites going up all the time. There's more field plots. The final thing that I should say is integration of data networks around the globe. So NEON has a standardized sampling design, which is great because we can take out the idiosyncrasies of each sample out of the equation because they've done them all the same. But if I try to compare this with the Smithsonian sites or with sites in Europe and other places, everyone has a different sampling design. And now we don't know what it's apples and oranges. And it's really hard to do this kind of large scale model, large scale integration. So we need more data. We need more integration. We need people to talk to each other. So we have consistent standards for field sampling and for remote sensing, especially that middle scale, which is going from the field, between the field and the satellite, which is airborne. So we need consistent airborne imaging as well. Cool, thanks. Yeah.
How big are those Jedi?
Yeah, so the Jedi footprint is a 25-meter diameter
For one pulse?
for one.
How do you ground truth?
So the Jedi Cal-Val team has people around the world that have been using airborne LiDAR from Gabon and French Guiana and US, Neon, all China, all these different sites, and they are they're doing calibration and validation based on that to be able to quantify their accuracy, and that accuracy extends in the vertical dimension. So how good are they getting that vertical profile, which is what LiDAR is supposed to do? But equally important, if not more important, is the horizontal accuracy, so the geolocational precision. That's a difficult and tricky subject. Right now, the newest product has a variation or a sigma of 10 meters, which is not great. But considering that we're doing these large scale analyses, let's say we run this all across Brazil, we have summary statistics, we can get a pretty good idea of what's going on in Brazil with that. But if we're trying to do fine scale analyses, that's going to be a problem. And there's ways to get around that. I mentioned Steve Hancock's bullseye method, which is something that we're doing, we're instituting. And what that essentially does is say if there is a bias in an orbit, it's likely systematic across the entire orbit. So we can take all the different sites and try to jiggle them in space and see where the correlation with the airborne LiDAR is best. And then we can make a systematic shift to the entire orbit. And that helps us get that geolocational accuracy from 10 to 1 or 2 meters. But that's very computationally intensive.
So how much does season impact developing these forest structure or the structure measurements?
Yeah, another great question. So everything here has been leaf on. So all the foliage, all the leaves are there, and so we can. It's a much better way to see that. Leaf off.
Do you have to be at like peak greenness, or like what if it's like early spring versus like?
Yeah. So yeah, neon is designed to be peak greenness. So they fly it at peak greenness because they're not just measuring structure. They're measuring all these other things. They want to make sure that they have the best sample, right? So that is peak greenness. G-light was in the fall, and so there were probably fewer leaves in the canopy, which might have biased. If you have full leaf on, then it's gonna be a lot more difficult to measure the structure because you're gonna be relying on those thin, skinny branches versus the big spread out leaves. But the good thing about fall was that that's the time that the leaves are changing. So different species have different biochemicals that at that period of time are expressing themselves. We're seeing the carotenoids and anthocyanins and these different chemicals that are much more active and noticeable versus everything is chlorophyll in the peak. And so that actually helped us. And we had several different flights and we looked at the difference. And we had like an early September all the way to a late October, which is really when the peak of the fall foliage. And that early October was the one where the hyperspectral diversity was telling us the most information about the distribution of those different species, which, back to your original question, really goes down to function. So yeah, so you can do it all, you know, I think you work with the data that you have, is the other thing. And you try to make it work. You try to get it to go as far as.
Well, and it'd be interesting to look at ecosystems that don't have such a strong seasonal signal. Like, yeah, the West Coast doesn't have carotenoids and
the tropics.
anthocyanins and, yeah,
Yep.
totally. Do we have any more questions? Miriam, go for it. I don't see anyone clamoring, so.
So can you tell me a little bit about conceptually how it works to have a laser pulse 25-meter diameter? Probably
Footprint on the ground.
footprint on the ground.
Yeah, so that's it. You can think of it as a kind of cone. And it's 25 meters by the time it hits the earth.
25 meters across the circle.
Yeah, the diameter of the circle.
That probably has multiple trees in it in most places, right?
Mhm.
So then we have like a mixed pixel, but it's not a pixel problem. How is that interpreted?
So I think it's interpretable in the sense that it doesn't matter if there's one tree or 10. What we're measuring is the forest structure in that footprint. And so they could be comprised of lots of different individuals or just one. The key data output is really what is the vertical distribution of the foliage in that footprint. If that makes sense. It's a waveform. And so we can actually see that, oh, well, it turns out that it's 42 meters at the top of the canopy in this forest. And there's a really strong mid-story and a very kind of empty ground story. And then we get that ground return or something like that. It's not telling us that there are how many individuals or what the species are or whatever. And so, you know, the major application for JEDI is characterizing biomass. And that's kind of what it does. And then this is it's kind of sub-applications for biodiversity. But it's explicit in call, but it's kind of secondary to biomass.
All right, let's thank our speaker one more time.